Good morning, everyone. Today we are going to complete our lesson about the light-independent reactions. You remember in the last lesson we explained the light-dependent reactions, and we said that by the end of the light-dependent reactions, reduced NADP and ATP are produced, and they will be used in the light-independent reactions. Light-independent reactions start with a very important process that we call it carbon fixation. Carbon fixation depends on fixation of carbon dioxide. This reaction happens inside the stroma in the chloroplast, unlike the light-dependent reaction which happens within the thylakoid membranes. Inside the stroma there are many enzymes that catalyze the processes happening here, and they said that the triose phosphate is the first stable carbohydrate sugar formed in the light-independent reaction. Triose phosphate is a 3-carbon sugar, while carbon dioxide is just 1 carbon sugar. Now we are going to learn how one molecule that has 1 carbon atom convert into a molecule which has 3 carbon atoms. The process of carbon fixation starts with ribulose biphosphate. Ribulose biphosphate is a 5-carbon sugar. It's going to fix carbon dioxide in a process that we call it carboxylation, which is adding carbon dioxide to the molecule. This reaction is catalyzed by an enzyme, we call it Rubisco. Rubisco stands for ribulose biphosphate carboxylase. Rubisco catalyzes the addition of carbon dioxide to ribulose biphosphate to form first a 6-carbon molecule which is unstable, quickly split into 2 molecules of glycerate 3-phosphate. So a 5-carbon sugar plus 1 carbon molecule which is carbon dioxide will form 6-carbon unstable molecule splits, giving 2 molecules of glycerate 3-phosphate. Each of them has 3 carbon atoms. Glycerate 3-phosphate cannot make sugar, or we cannot call it sugar. Why? Because here we added carbon dioxide. When you add carbon dioxide, you interrupt the ratio that should be present in any carbohydrate. Any carbohydrate, as you know, has the formula CnH2nOn. So the ratio between hydrogen and oxygen in any sugar should be 2:1. Here we added carbon dioxide, so we added oxygen without adding hydrogen. So in order to make a sugar, we need to add more hydrogen. What is the source of this hydrogen? The source of hydrogen is reduced NADP, which is produced from the light-dependent reaction. Reduced NADP is going to donate its a pair of protons to glycerate 3-phosphate, and then NADP will be a reduced NADP. An oxidized NADP. This process requires energy. The source of energy is ATP. So also which is produced from the light-dependent reaction. So both ATP and reduced NADP catalyze the conversion of glycerate 3-phosphate into a sugar molecule, which has the normal molecular formula of sugar, which is triose phosphate. Triose phosphate is a sugar molecule, and it is we consider the first stable molecule formed in photosynthesis process. Suppose we have 5 molecules of triose phosphates. Each one of them has 3 carbon sugars, 3 carbons. So 5 into 3, so 5 triose phosphates have 15 carbon atoms. So how many ribulose biphosphate they can regenerate? Remember, ribulose biphosphate has 5 carbons. So 15 carbons in triose phosphate can make 3 molecules of ribulose biphosphate. This process also requires energy, which is sourced by the ATP. So 3 ATP molecules are used to convert 5 molecules of ATP into 3 molecules of ribulose biphosphate. But here, in this way, triose phosphate will regenerate the ribulose biphosphate in what we call Calvin cycle. Calvin cycle is named after the scientist Melvin Calvin, who discovered the scheme of work in this process, and he won the Nobel Prize for his discovery. Let's summarize the Calvin cycle. Calvin cycle starts with carbon fixation, where the ribulose biphosphate reacts with carbon dioxide in presence of Rubisco to form the 6-carbon unstable molecule, splitting, giving 2 molecules of glycerate 3-phosphate. And then glycerate 3-phosphate in presence of energy from ATP and hydrogen on NADPH2, they give molecules, each glycerate phosphate will give 1 molecule of triose phosphate. So if we started with 3 molecules of ribulose biphosphate. Each of them has 5 carbons, means that we have here 15 carbon molecules. They are by enzyme Rubisco, they will give glycerate 3-phosphate. How many glycerate 3-phosphate they can produce? If I have here 15, so we expect that we can have here 5 molecules of glycerate 3-phosphate. They use ATP and reduced NADP to form triose phosphates. Triose phosphates is 3-carbon sugar. In order to regenerate back ribulose biphosphate, so 2 carbon atoms should be added. So we are going to use 2 more carbon dioxide molecule per each triose phosphate to regenerate ribulose biphosphate, which is 5-carbon sugar. This process also requires energy, which is supplied by the ATP. But where is the sugar that we need for our food and for the photosynthesis process? It was found that most of the triose phosphate is used to regenerate the ribulose biphosphate to replenish the Calvin cycle, so the cycle can continue and we can make more and more of triose phosphate. And a very small amount only of triose phosphate is used to build up hexose. So very small, if we have 12 triose phosphates, if we have 12 molecules of triose phosphate, 10 of them will be used to regenerate ribulose biphosphate, while only 2 molecules will be used to make hexose phosphate. Remember that hexose phosphate is 6-carbon sugar, so it does require 2 molecules of triose phosphates. Okay, so out of 12 triose phosphates, only 2 are used to form hexose phosphate, and then they undergo condensation reaction to form starch and the other complex sugar molecules. Let's see here this part. So if we want to summarize the role of reduced NADP and ATP in the Calvin cycle, what can we say? NADP, reduced NADP, reduces glycerate 3-phosphate to form triose phosphate. They are used here in this process, while the ATP provide energy which is required for this reaction. Okay, now we summarized the light independent reaction that doesn't need light. It is enzyme dependent, so it depends on the temperature and availability of ribulose biphosphate. As long as ribulose biphosphate is available, the cycle can continue. Okay, inshallah, in the next lesson we are going to study the experiments related to the photosynthesis process. Thank you very much.